Now let's take a more close look at the lateral surface of the human brain. And what we're looking at here is the right hemisphere of a brain specimen. And to begin with, let's find the central sulcus. The central sulcus is a long, roughly straight sulcus, except for a distinctive S-shaped bend that represents the hand in the somatic motor and somatic sensory cortex. But otherwise, the sulcus extends from the dorsal midline to the lateral fissure in roughly a straight trajectory. All of the cerebral hemisphere to the front of that central sulcus is the frontal lobe. And the frontal lobe is comprised of a precentral gyrus and then three gyral structures that run more or less in the long axis of the hemisphere. Near the dorsal midline is the superior frontal gyrus. Near the middle of this frontal lobe is the middle frontal gyrus. And then along the inferior margin of the frontal lobe is the inferior frontal gyrus. So again, superior, middle, and inferior frontal gyri that are separated by two meandering sulci, an inferior frontal sulcus and a superior frontal sulcus. Now let's look behind the central sulcus at the parietal lobe. So again, here's the central sulcus, and the posterior boundary of the central sulcus is the long, mostly straight post-central gyrus. And then just behind that post-central gyrus, there are two gyral formations. Now, there are subdivisions that we could name, but for our purposes, we'll keep it simple and talk about these two structures as an inferior parietal lobule in this region of the parietal lobe, and then a superior parietal lobule, which is up here along the dorsal midline of the hemisphere. And between the superior and inferior parietal lobule is a deep sulcus of the parietal lobe called the intraparietal sulcus. And that sulcus runs in the long axis of the hemisphere, approaching the central sulcus at nearly a right angle. Now, just behind the parietal lobe is the occipital lobe. And it's somewhat arbitrary how we define the boundary between the occipital lobe and the parietal lobe. It almost doesn't make sense to do so based on the function of this part of the cerebral cortex. But based on the anatomy, as we view the lateral surface, it is helpful to define that boundary. There's a deep sulcus that's more obvious along the midline that comes out to the lateral surface of the brain in this location. That's the parieto-occipital sulcus. So if we can draw an imaginary line from that parieto-occipital sulcus down to a notch formed by the way the dura tends to restrict the development of the inferior surface of the occipital lobe, we have a boundary that roughly divides the occipital lobe from the parietal lobe. This notch that we can see right about in this region, that's called the preoccipital notch because it helps to define the anterior boundary of the occipital lobe. And now let's go inferior to the lateral fissure, which is located right here, and explore the features of the temporal lobe. From the lateral surface of the hemisphere, we see three long, straight, parallel gyri, known as the superior, the middle, and the inferior temporal gyri. I want you to notice that this arrangement of three long, straight, parallel gyri is quite similar to what we found in the frontal lobe. So between these three gyri are two sulci that are more or less straight and parallel. There's the superior temporal sulcus between the superior and the middle temporal gyri, and then the inferior temporal sulcus between the middle and the inferior temporal gyri. To get a better look at the inferior temporal gyrus, we'll need to angle the temporal lobe just a bit so you can see the lateral surface curving into the ventral aspect of the brain. We'll see the rest of the gyri and sulci of the temporal lobe when we explore the ventral view of the brain in another video lesson. Now, if we were to compare one hemisphere to the other, we're looking now at the right hemisphere, and if we looked at the left hemisphere, we would see all of those same gyral features, although there may be a few aspects of this lateral view that could be a bit different. One of them concerns this deep fissure called the lateral fissure. This lateral fissure tends to be longer and straighter in the left hemisphere compared to the right. And this, we think, accommodates some of the specialized regions of the posterior temporal lobe and inferior parietal lobule that have to do with processing human language. So in most of us, our lateral fissure is longer and straighter in the left hemisphere as compared to the right hemisphere, where that fissure often takes a sharp upward bend rather than extending further back in more of a straight line. So that's a brief tour of the lateral surface of the human brain. Now let's explore the medial surface of the human brain using a specimen that's been cut right down the midline in the mid-sagittal plane. To reorient you to this view of the brain, let's again identify the embryological divisions that are visible in this view of the brain. Most of the cerebral cortex that we see, as well as now the cut interior surface of the forebrain, is derived from the prosencephalon. The cerebral cortex is telencephalon, and then now what we can see here is the diencephalon, being comprised of a region called the thalamus, and then just below the hypothalamus. Together, the diencephalon and the telencephalon are what have become of that first anterior bulge of that developing neural tube that we call the forebrain. From about this location on back, this is what we call the hindbrain, and that includes much of the brainstem and the cerebellum. Between the hindbrain and the forebrain is a small division of the developing neural tube that became a brain region called the middle brain, or the mesencephalon, and that's what we see right here. So we have forebrain, middle brain, and then the hindbrain, which in this view we can recognize as the pons and cerebellum together forming the metencephalon, and then down below is the medulla oblongata, or the myelencephalon. Now let's look again at the forebrain and talk about the gyral structures that we can see in this view of the telencephalon. So just on top of the diencephalon is this broad band of white matter called the corpus callosum. And that is a very obvious visual feature that helps define the contour of this medial set of gyri that sit above it. Right above the corpus callosum is a long gyrus called the cingulate gyrus. And this gyrus has many divisions that we could identify, but we'll keep it simple and just call this the cingulate gyrus. Just above the cingulate gyrus is a sulcus called the cingulate sulcus. And then superior to the cingulate sulcus is the superior frontal gyrus that forms the dorsal margin of the frontal lobe. The superior frontal gyrus extends back towards this region around the medial terminus of the central sulcus called the paracentral lobule. The paracentral lobule is bounded below by the cingulate sulcus, and then posteriorly by the marginal branch of the cingulate sulcus. So this then is the paracentral lobule, around the termination of the central sulcus. So just posterior to that central sulcus then is the parietal lobe. And just behind the marginal branch of the cingulate sulcus is a gyral structure called the precuneus. The precuneus is the boundary of the parietal lobe and the occipital lobe. Between those two lobes is a long, straight sulcus that runs from the inferior surface of the hemisphere all the way up to the dorsal midline. This sulcus is called the parieto-occipital sulcus, and it's the posterior boundary of the precuneus. Now, running in most brains almost at a right angle to this parieto-occipital sulcus is the calcarine sulcus. And the calcarine sulcus is present right here. The calcarine sulcus is an important landmark that identifies the location of the visual parts of the brain on the medial surface of the occipital lobe. Down below the calcarine sulcus is the lingual gyrus, and above the lingual gyrus is the cuneus. The lingual gyrus forms something like a tongue on the bottom of the occipital lobe, and lingual means tongue. Just above that calcarine sulcus and behind the parieto-occipital sulcus is this wedge shape that we call the cuneus, and cuneus is a word that means wedge. So we have the tongue, the wedge above the tongue, and then the gyral formation, again just in front of the wedge, is called the precuneus, or the structure in front of the wedge. We'll come back and talk more about the structures that are visible here in the brainstem in another lab lesson. But for now, again, just to quickly review, we have the forebrain, including the telencephalon and the diencephalon, the midbrain, or the mesencephalon, and then below the middle brain is the rest of the brainstem, including the pons, the cerebellum, and the medulla oblongata. One of the most important sulci in the human brain is the central sulcus. And one might imagine when we look at the human brain that it ought to be easy to determine which one is in fact the central sulcus. Sulci, of course, are the term we use for the spaces that we see in the surface of the cerebral hemisphere. So one interesting point about the central sulcus is that sometimes it can be very difficult to find in a human brain because the brains often don't look like the simplified figures we often see in textbooks. But there is a way to find the central sulcus that's foolproof. And to do so, I have to look at a specimen that's been cut right down the midline along the mid-sagittal plane. So I'm going to put aside our whole brain and show you a hemisphere that's been sectioned in the mid-sagittal plane. So here we have a hemisphere, and this broad structure of white matter here is called the corpus callosum. And just above that corpus callosum is a gyrus called the cingulate gyrus. And if you would, I want you to appreciate the sulcus that forms a space just on top of the cingulate gyrus. This is called, conveniently enough, the cingulate sulcus. So we follow this sulcus back from the front of the brain towards the back of the brain, and we find that just past the middle of the hemisphere, there's a point where this sulcus makes a sharp upward bend. That's called the marginal branch of the cingulate sulcus. Now, if we identify that marginal branch of the cingulate sulcus and look for the very first sulcus that intersects the midline, I know that this will always be the central sulcus. So I'm going to just gently put my wooden stick in that space and then turn the brain around and we'll see if this is in fact the central sulcus. So now, as we look at the lateral surface of the brain, we've identified the central sulcus. And the other features that help us know for certain that it's a central sulcus are the fact that it's a long sulcus that makes a lazy S-shaped bend on its way from the dorsal midline down towards the lateral fissure, which is a large space separating the temporal lobe and the frontal and parietal lobe. Now, to see the central sulcus even better, I'm going to gently pull away some of this tissue that's covering the surface of the brain. This is called arachnoid, and the arachnoid is a tissue that helps to create a buffering space over the surface of the cerebral cortex. Underneath the arachnoid in life is perhaps a millimeter or two of cerebral spinal fluid. So as I tease apart this tissue, we can see the full length now of the central sulcus, all the way from its medial terminus along the dorsal midline to its lateral terminus, very near the lateral fissure. It's worth noting how the body is mapped out along the banks of this sulcus. The gyrus that forms the anterior bank is called the precentral gyrus, and this contains the motor map of the contralateral body. The gyrus that forms the postcentral structure is called the postcentral gyrus, and it contains the somatic sensory map of the contralateral body. And there's a beautiful mapping of the body from the midline to the lateral fissure, beginning along the medial surface of the brain, where the central sulcus intersects the dorsal midline of the hemisphere. This is a structure where the precentral and postcentral gyri come together to form a lobule, which is just a word that means a larger gyral structure. This lobule is called the paracentral lobule because it contains the medial termination of the central sulcus. The paracentral lobule is where we have the representation of the contralateral foot. This is the right hemisphere, so this is where the left foot would be represented for motor control and for somatic sensation. And as we progress from the paracentral lobule onto the dorsal and lateral surface of the brain, we ascend that body map from the leg up towards the hip region and then into the trunk. And then just where we see this distinctive S-shaped bend in the central sulcus, that's where we know that our body map is progressing from the trunk to the forearm and then eventually into the region that represents the hand. Where we see this prominent S shape in the central sulcus, that's the representation of the contralateral hand, again with motor control being mapped to the precentral gyrus and somatic sensation in the postcentral gyrus. Now, this body map is exquisite, and it's a map that perhaps not Michelangelo would have depicted, but perhaps Pablo Picasso, since there's a bit of a cubistic break in the human form along the central sulcus just below this S shape. Rather than continuing on with the representation of the hand and the digits, just below this S shape we have now representation of the face. So that the face now is represented for motor control in the precentral gyrus and somatic sensation in the postcentral gyrus just below the tips of the fingers. And the representation of the face carries on down to the lateral termination of the central sulcus. For this lab lesson, we want to explore the ventral surface of the human brain. So I'm going to take this specimen and hold it up so that you can have a clear view of the ventral surface of the brain. And what I'd like to do is just walk you through the features of this brain surface from the front part, the anterior part of the brain, through the back part, the posterior part. So towards the front part of the brain, the first set of structures that we can note are these two long tracts on either side of the midline that sit on the bottom of the frontal lobe. These are the lateral olfactory tracts that terminate in an olfactory bulb. And as those names imply, these structures have to do with our sense of olfaction or our sense of smell. The next interesting feature we encounter is the optic nerve from one side and the other that come together and form an X shape. That X shape is called the optic chiasm. Chiasm means an X or a cross, so it's a crossing of the nerve fibers that form these two optic nerves. Just behind the optic nerves is the floor of the diencephalon, or the ventral surface of the hypothalamus. And there's a bit of a hole in this specimen, which is where the stalk of the pituitary attaches to the hypothalamus. That stalk was removed, and in its place we see a little bit of a hole that allows us to look through into a space that sits right in the middle of the hypothalamus called the third ventricle. Just behind the stalk of the pituitary are two bumps that define the posterior limit of the hypothalamus. Those are called the mammillary bodies, which are seen right here. Now, if I tilt this specimen a little bit more, you can appreciate a dark space just behind these mammillary bodies. And this is roughly the junction of the diencephalon and the midbrain. In fact, the space is defined by these two broad columns called the cerebral peduncles, or cerebral stalks, one on either side of this space. Those stalks are a component of the midbrain, or the mesencephalon. And as the term peduncle or stalk implies, this is a massive bundle of white matter that allows the forebrain to connect with everything below it, the hindbrain and the spinal cord. Now, just inferior to the cerebral peduncles, as we see them here in the midbrain, is the pons and the cerebellum. The pons extends out laterally, again another massive bundle of white matter connecting the brainstem to the cerebellum. And just inferior to the pons, we have the medulla oblongata, or the medulla for short. And on the ventral view of the medulla, two striking features appear along the midline. It's a pair of structures called the medullary pyramids that run on either side of the midline. This is a very important fiber pathway that runs through the brainstem. It's called the cortical spinal tract, and there's one on each side of the midline. These tracts contain the fibers of the motor cortex that are sending their axons down into the spinal cord to govern voluntary movement. One additional feature of the medulla that sits just next to the medullary pyramid is this bulging shape. It looks like an olive that has been cut in half and stuck to the side of the medulla, just lateral to the medullary pyramid. And thankfully, that's what we call this structure. This structure is called the olive, so it's easy to remember. So we have the medullary pyramids right along the midline, and then just lateral to the medullary pyramid is this bulging structure called the olive. Now, let's turn our attention to the cerebral cortex as we can see it on the inferior surface of the brain. So what we see towards the front of the brain is the inferior surface of the frontal lobe. And this part of the frontal lobe sits right above the orbits in the skull, and for that reason we call this the orbital cortex. Just near the posterior part of the frontal lobe, we begin to encounter the temporal lobe, which grows out and underneath the frontal portion of the lateral fissure. So in this part of the cortex, we're looking at the inferior surface of the temporal lobe. But to see the rest of the inferior surface of the temporal lobe, we need to move to a different specimen that has had the brainstem and the cerebellum removed. And now in a second specimen, we can have a more clear view of the entire ventral surface of the brain with the brainstem and cerebellum removed, so that we can see the features on the cerebral cortex of the inferior temporal and occipital lobes. So here again is the temporal lobe that extends from the posterior part of the frontal lobe all the way back towards the occipital lobe, with the junction being somewhere in this region, right about here. So this bit of brain is the temporal lobe. And on its inferior surface, we have a long gyrus that extends throughout the length of the inferior temporal lobe back towards the occipital lobe. That's called the occipital-temporal gyrus. Sometimes there's a small sulcus that divides it into a lateral and medial components. And then along the medial edge of the temporal lobe, there's a structure that we call the parahippocampal gyrus. And deep in the anterior part of the temporal lobe, in this position, this parahippocampal gyrus folds in upon itself into a structure that we call the hippocampus. And we'll see the hippocampus when we look at sections through the brain. Now the occipital-temporal gyrus and the parahippocampal gyrus extend down into the occipital lobe. And at some point in the occipital lobe, we define a region that is a boundary between the lingual gyrus near the midline and what remains here of the occipital-temporal gyrus. Sometimes this region of the inferior occipital lobe is called the fusiform gyrus because of the superficial shape that is sometimes appreciated as these sulci come together. We see similar features in the opposite hemisphere, the same set of long, lateral, and medial occipital-temporal gyri, and then along the midline of the temporal lobe, the parahippocampal gyrus. My goal for this lab lesson today is to help you conceptualize the gyri and the sulci that define the surface features of the human brain. When we look at the adult human brain, it's hard to appreciate what is consistent from one specimen to the next and what's different. This lesson is all about the consistencies, and once you conceptualize those consistencies, it will be just a wonderful experience to appreciate what's different from one brain to the next. When you look at the surface features of the brain, the gyri and the sulci, we see patterns, and these patterns are analogous to the fingerprints on each of your digits. Those fingerprints have standard patterns for each digit, but in each individual, of course, we know that our fingerprints are quite distinct. Something similar can be said of the surface features of the cerebral hemispheres. There are, in fact, primary sulci and gyri that are present in every normal human brain. And then there are secondary and tertiary gyri that help define those distinctive qualities of the surface pattern in each cerebral hemisphere. So we're going to focus on those primary sulci and gyri. And I think a great way to conceptualize the organization of the cerebral hemispheres will be to actually allow you to see how I place my fingers on the gyri of the cerebral hemisphere. And I want you to imagine that my fingers are gyri and the space between my fingers are sulci. So let's begin with the central sulcus. The central sulcus is right near the middle of the hemisphere, and it's defined by two roughly parallel gyri, a precentral gyrus and a postcentral gyrus. So my index finger is representing and overlying the precentral gyrus. My middle finger is overlying the postcentral gyrus, and the space between my fingers represents the central sulcus. Now, while I keep those two fingers there, I want to demonstrate for you the remaining gyral features of the frontal lobe. And to do that, what I'm going to do is place three fingers in parallel that are roughly orthogonal to this central sulcus. So I have a finger down below, which is overlying the inferior frontal gyrus, a finger in the middle that's overlying the middle frontal gyrus, and then a finger up on the top of the hemisphere that's representing the superior frontal gyrus. So three fingers representing three roughly parallel gyri that are meeting the central sulcus almost at a right angle, a little bit more obtuse than a right angle. So now let's look at the parietal lobe. I'll reestablish the central sulcus with my left hand. So now my middle finger is representing the precentral gyrus, and my index finger is representing the postcentral gyrus. And to conceptualize the parietal lobe, all I need to do is place two fingers that are running, again, roughly at a right angle to the central sulcus. My index finger is near the dorsal margin of the hemisphere. We call that gyral formation the superior parietal lobule. And my middle finger is just below the superior parietal lobule, representing the inferior parietal lobule. The space between them is a prominent sulcus of the parietal lobe called the intraparietal sulcus. Okay, now let's move to the temporal lobe. In the temporal lobe, just as we did for the frontal lobe, we can conceptualize this lateral surface as being comprised of three long parallel gyri. So we had three long parallel gyri in the frontal lobe, and we have three long parallel gyri in the temporal lobe. We have a superior temporal gyrus, a middle temporal gyrus, and an inferior temporal gyrus. And the two spaces between my three fingers would be the superior temporal sulcus and the inferior temporal sulcus. Now with this specimen, the brainstem and the cerebellum are preventing me from conceptualizing the inferior surface of the brain, so I'm going to opt for an additional specimen to extend the placement of my fingers to the inferior surface of the temporal lobe. In this second specimen, I've removed the brainstem and the cerebellum so that we can have a better view of the inferior surface of the cerebral hemisphere. Now, I just showed you placing three fingers along the lateral surface of the temporal lobe, and then to complete the picture of the temporal lobe, I'm just going to lay my additional two fingers right along two long gyri that we see on this inferior surface. So now I've got five fingers on the temporal lobe. On the lateral aspect, I've got a superior temporal gyrus, a middle temporal gyrus, and an inferior temporal gyrus. And then as we look onto the ventral and medial aspect of the temporal lobe, my index finger is representing a long straight gyrus that runs from the temporal lobe back towards the occipital lobe. This is called the occipitotemporal gyrus, and sometimes the posterior part is called the fusiform gyrus. And then my thumb represents a very interesting medial gyrus of the temporal lobe called the parahippocampal gyrus. In fact, the knuckle of my thumb is sort of sticking out just a little bit because indeed there's something that looks like a knuckle on this gyrus called the uncus. The parahippocampal gyrus folds in upon itself to form a really wonderful structure in the brain called the hippocampus. And we'll see that in cross-sections through the brain. So 5 fingers on 5 gyri in the temporal lobe. And now to complete our lesson on placing fingers on the gyri of the cerebral hemispheres, I've got yet a third specimen that allows me to work on the medial surface of the hemisphere. But to approach the medial surface, I want to begin again with the central sulcus. And you'll recall that I defined the central sulcus with 2 fingers on the lateral surface, with the precentral gyrus, a postcentral gyrus, and the space between my fingers being conceptualized as the central sulcus. Now what I want to do is I want to reverse my approach to the central sulcus and let my fingertips wrap along the dorsal surface of the brain such that my fingertips come together in the part of the brain called the paracentral lobule. So now imagine that my 2 fingers are fused together. That's the relation of the pre and the postcentral gyri to the paracentral lobule. So that portion contributed now by my middle finger is the precentral gyrus, and the part contributed by my index finger is the postcentral gyrus. And again, those 2 fingertips or 2 gyri will fuse just above and in front of the branches of the cingulate sulcus. Now the rest of the frontal lobe can be conceptualized as having 2 long gyri. The cingulate gyrus that runs right on top of the corpus callosum, and then the gyrus that forms the dorsal margin of the hemisphere. This is simply the continuation of the superior frontal gyrus onto the medial face of the hemisphere. Now let's look at the occipital lobe. The occipital lobe is in this region of the brain, and it can be conceptualized as having 2 gyri that are somewhat parallel to each other. The inferior of the 2 is called the lingual gyrus, and this is a gyrus that forms something like a tongue shape on the bottom of the occipital lobe. Lingual means tongue. And then just above the lingual gyrus is a wedge-shaped structure called the cuneus gyrus. Cuneus is a Latin word that means wedge. Now just in front of the wedge is a gyrus that wraps around from the superior parietal lobule, and this we call the precuneus, which of course would simply mean in front of the wedge. So the rest of the medial face then is comprised of an occipital lobe defined by 2 gyri, a lingual gyrus and a cuneus gyrus, with a space between them being the calcarine sulcus. And then at about a right angle is the precuneus gyrus, with the space between the precuneus gyrus and my 2 fingertips, representing the occipital lobe, is a long straight sulcus called the parietal occipital sulcus. Now you may not have a human brain to wrap around your fingers, but if you're fortunate enough to have access to a brain model, I would encourage you to do this exercise. Perhaps you can even make your own brain model out of modeling clay, or perhaps even just with a round-shaped balloon that you can mark up and put your own fingers on the gyri to help conceptualize the complex anatomy of the cerebral hemispheres. Hello everyone, welcome back to Medical Neuroscience, and welcome to this brief review of the surface anatomy of the brainstem. This is an attempt to break down some of the complexity of the human brain, and so my goal is to help you appreciate that complexity, but also give you a framework for coming to grips with it. So there's actually nothing new in this tutorial. Rather, I want to put before you again the surface of the brainstem, because we will be diving shortly into the complexity of the cranial nerves and the nuclei in the brainstem to which they connect. And I think in order to really get the most out of the tutorials that you are about to view on these subjects, it will be really important for you to be able to describe the surface features of the midbrain, the pons, and the medulla oblongata. So what I want to do is show you again the surface of the human brainstem. Here's a model of the brainstem with parts of the deep forebrain nuclei attached on the top, and I want you to be able to review the surface features of the midbrain, the pons, and the medulla as seen from the ventral view, as well as from the dorsal view as best we can. So what I'm going to do is give you another look at the actual human brainstem and give you a chance just to refresh in your mind what are the principal features that define each of these subdivisions, such as the cerebral peduncles of the midbrain or the medullary pyramids of the medulla oblongata. So I hope you enjoy this review, and I hope it sets the stage for a productive viewing of the remaining tutorials that pertain to the way that the cranial nerves connect with this complex part of the human brain. So I'll see you next in the lab. For this lab lesson, we want to explore the ventral surface of the human brain. So I'm going to take this specimen and hold it up so that you can have a clear view of the ventral surface of the brain. Now, if I tilt this specimen a little bit more, you can appreciate a dark space just behind these mammillary bodies. And this is roughly the junction of the diencephalon and the midbrain. In fact, the space is defined by these two broad columns called the cerebral peduncles, or cerebral stalks, one on either side of this space. Those stalks are a component of the midbrain, or the mesencephalon. And as the term peduncle or stalk implies, this is a massive bundle of white matter that allows the forebrain to connect with everything below it, the hindbrain and the spinal cord. Now, just inferior to the cerebral peduncles, as we see them here in the midbrain, is the pons and the cerebellum. The pons extends out laterally, again another massive bundle of white matter connecting the brainstem to the cerebellum. And just inferior to the pons, we have the medulla oblongata, or the medulla for short. And on the ventral view of the medulla, two striking features appear along the midline. It's a pair of structures called the medullary pyramids that run on either side of the midline. This is a very important fiber pathway that runs through the brainstem. It's called the cortical spinal tract, and there's one on each side of the midline. These tracts contain the fibers of the motor cortex that are sending their axons down into the spinal cord to govern voluntary movement. One additional feature of the medulla that sits just next to the medullary pyramid is this bulging shape. It looks like an olive that has been cut in half and stuck to the side of the medulla, just lateral to the medullary pyramid. And thankfully, that's what we call this structure. This structure is called the olive, so it's easy to remember. So we have the medullary pyramids right along the midline, and then just lateral to the medullary pyramid is this bulging structure called the olive. In this lesson, we're going to discuss the means by which blood is supplied to the human brain. The blood supply to the brain comes from below. So in order to inspect those vessels, we need to look at the inferior surface of the brain. Blood is supplied to the brain through two major pairs of arteries. Towards the anterior side of the brain, those arteries are the internal carotid arteries. Here's the left internal carotid artery, and then here is the right internal carotid artery. These are the two major arteries that supply what we call the anterior circulation. The posterior part of the brain and much of the spinal cord is supplied by a different pair of arteries. These are called the vertebral arteries. Here is the cut end of the right vertebral artery, and right next to it is the cut end of the left vertebral artery. the vertebral arteries contribute what we call the posterior circulation. So let's talk about these two major circulations to the brain, beginning with the posterior circulation. These vertebral arteries arise and enter the cranium, and then they join together to form a single midline artery called the basilar artery. And that's what we see right about the junction of the medulla and the pons, is where the two vertebral arteries fuse. And then the basilar artery runs up along the length of the pons towards the midbrain. Now, along this length of this posterior supply, there are branches that supply the brainstem and the cerebellum. Just below this cut end of the vertebral artery is a junction of a branch right here called the posterior inferior cerebellar artery. So this artery, which is cut in this specimen, attached to this right vertebral artery. The posterior inferior cerebellar artery supplies, as its name applies, the posterior and inferior aspect of the cerebellar hemisphere, as well as giving rise to branches that supply the lateral part of the medulla. Now, typically, just after the junction of the vertebral arteries to form the basilar artery, we have two additional arteries that supply the brainstem and the inferior part of the cerebellum. These are called the anterior inferior cerebellar arteries. And here we see them on either side of the lower portion of the basilar trunk. So these, again, are the anterior inferior cerebellar arteries. Now, near the top of the pons, where the basilar artery is about to split and form two major cerebral arteries, there are two branches that supply the superior surface of the cerebellum. These are the superior cerebellar arteries. Here is the left superior cerebellar artery, and then over here, I think you have a better view of this, there's the right superior cerebellar artery. Now, as these arteries wrap around the brainstem, they give rise to short branches that supply the lateral and dorsal aspects of the midbrain and the pons. Now, along the length of this basilar artery are numerous short branches that supply the interior structures here within the pons. And these vessels are very small, very fine, and sometimes they clot and produce strokes in the brainstem that can have a variety of consequences depending upon what structures are injured. Now, let's try to look deep into this central region on the ventral surface of the brain and see what happens to that basilar artery. The basilar artery, just anterior to the superior cerebellar arteries, splits into two parts. One branch goes to form the posterior cerebral artery in one side, and then the opposite branch forms the posterior cerebral artery on the other. Now, in order to get a better view of those cerebral arteries, we need to look at a specimen where the brainstem and the cerebellum has been removed. Here's a specimen where the brainstem and cerebellum has been removed, so we can see what happens to the posterior cerebral artery at the top of the basilar trunk. What we see here are blood vessels that extend along the inferior surface of the temporal and occipital lobe, and then along the medial surface of the occipital and temporal lobes here. These are branches of the posterior cerebral artery. And right at the junction of the basilar and it's split into a left and right posterior cerebral artery, there are small branches that supply the adjacent structures here in the midbrain. Well, that concludes our survey of the posterior circulation. Now let's look at how the posterior circulation meets the anterior circulation. So to do that, we need to go back to the specimen where we have a clear view of the blood vessels on the ventral surface of the brain stem and the diencephalon. Now we've got our first specimen in hand again, And we want to follow the posterior circulation to its junction with the anterior circulation. So again, the posterior circulation arises from the two vertebral arteries that join to form the basilar artery, which then runs along the midline of the pons, and in the region of the midbrain, that basilar artery splits into two posterior cerebral arteries that supply the inferior surface of the temporal and occipital lobes. Now, there are short communicating arteries that connect that posterior circulation to the anterior circulation, and we can see them right here. In this brain, they're actually quite prominent. This is called the posterior communicating artery, and it connects the posterior cerebral to the internal carotid. So there we have our right posterior communicating artery, and then on the opposite side we have our left posterior communicating artery. In this brain, these arteries are quite symmetrical, but it's not unusual to find a marked asymmetry in the dimensions of these two posterior communicating arteries. Okay, so this is the means by which the posterior circulation meets the anterior circulation, which is derived from our internal carotid arteries. Here's our left internal carotid and our right internal carotid. So now let's discuss the anterior circulation. These two carotid arteries give rise to two large branches. One branch runs in the anterior direction, just on top of the optic chiasm, and that forms an artery called the anterior cerebral artery. The anterior cerebral artery begins right there at the tip of my pointer, and it continues along the midline of the hemisphere. And if I gently separate these two hemispheres, we might be able to see the junction of the anterior cerebral artery and the midline of the hemisphere. And what happens here is that there are two anterior cerebral arteries, one that gives supply to the left hemisphere, one that gives rise to the right hemisphere. And if I gently reflect away the optic chiasm, I think we can see those arteries as they come together. And there is a short anterior communicating artery that connects the two anterior cerebrals. So just in front of my pointer, we see just a small little touch of an artery that allows the two anterior cerebrals to communicate. Now, to really see the anterior cerebral artery, what we want to do is we want to look at the mid-sagittal plane through the hemisphere and see that artery as it supplies the medial face of the hemisphere. So now that we have a brain that's been sectioned in the mid-sagittal plane, we can see what happened to that anterior cerebral artery. So here's the anterior cerebral artery, right where it is coming off the internal carotid. And we see that this anterior cerebral artery gives rise to branches that wrap around the cerebral cortex on top of the corpus callosum, supplying this cingulate gyrus and the superior frontal gyrus, all the way back through the region of the paracentral lobule. Now, these arteries have been cut right about in this position, so we can't follow them all the way back, but I think we can see very nicely how this artery hugs the medial surface of the hemisphere as it supplies the frontal lobe and the paracentral lobule. In addition to the anterior cerebral artery, the internal carotid gives rise to one more major arterial supply to the brain, and that's the middle cerebral artery. In some hemispheres, it appears as though the internal carotid just keeps on going, penetrating through this space between the temporal lobe and the frontal lobe. That space is the lateral fissure. And indeed, once we get beyond the junction of the anterior cerebral artery, the internal carotid is known as the middle cerebral artery. And this artery runs through that lateral fissure, supplying blood to the temporal lobe and the inferior surface of the frontal lobe, and emerges on the lateral surface of the brain. And underneath this arachnoid tissue, we can see branches of the middle cerebral artery as it supplies roughly the middle central portion of the cerebral hemisphere. So all of this cortical region, including the central part of the parietal and frontal lobe and the superior part of the temporal lobe, is supplied by branches of the middle cerebral artery. So far, what we've discussed is the arterial supply to the brain. But to complete the story, we have to talk about the venous drainage of blood to the brain. And for that, we want to inspect a sample of dura mater. This is the tough tissue that surrounds the outside of the brain. In fact, dura mater is one of those great words in brain anatomy that is exactly what it means. Dura mater means tough mother. So this tissue is a very tough, leathery sort of protective covering on the outside of the brain surface. However, in certain places, this tissue separates into two layers that form a venous sinus that allows blood to drain out of the brain. One such venous sinus is right along the midline here. So imagine this bit of tissue sitting around the brain such that the space between the hemispheres would be right here. So this sinus is called the superior sagittal sinus. And in fact, on the back part of this dura, we've made an opening so that we can see a bit of that sinus space. In fact, I can put the wooden stick right into that sinus so that you can see that there is indeed a bit of a triangular shaped channel right along the midline of this tissue that allows blood to drain from the cerebral hemispheres. That blood then meets another sinus space, which is in the back part of the cranium, right in this region. This is called the confluence of sinuses because the superior sagittal sinus and two other minor sinus systems drain into this receptacle, and from there the blood flows transversely through another space within this dura called the transverse sinus. And again, I can put my probe through there to demonstrate this space through which blood is leaving the cranium. So this transverse sinus then forms a structure called the sigmoid sinus, which then exits the skull through the jugular foramen as the jugular vein. And by this system of dural sinuses, blood is drained from the brain and returned back to the venous vascular system, which enters then the heart and is once again circulated through the pulmonary vessels. One other point about the dura, well, it's in my hand. This is the outer surface of the dura, and here's that superior sagittal sinus. If we look on the inside, we see a flap of tissue called the falx cerebri, which forms a bit of a separation between the two hemispheres. So this bit of dura sits right between the left and right cerebral hemispheres, with this inferior margin of the falx being a small narrow sinus that allows blood to drain from deep within the brain. And so this bit of tissue sits right on top of the corpus callosum. Hello everyone, welcome again to my backyard. I'd like to spend just a few minutes with you at this time talking to you about the cranial nerves and the spinal nerves. And this will set up a tour of the cranial nerves that I'd like to show you in the laboratory with a real human brain. Well, my learning objectives for you are, first, I want you to be able to discuss the surface features of each of the three subdivisions of the brainstem. And so in order to understand how the cranial nerves fit in, you need to know something about the divisions of the brainstem. And as you become more and more familiar with both the brainstem and the cranial nerves, I really want you to accomplish this learning objective. I want you to be able to sketch the relationships of the cranial nerves, specifically nerves 3 through 12, to the three subdivisions of the brainstem. So that may mean that you need to find a large piece of paper, a whiteboard, a chalkboard, maybe even just some dirt and a stick where you can make some sketches. Wouldn't that be fun to draw pictures of the brain in the dirt at your feet? But whatever it takes, I would encourage you to make visible your knowledge and your understanding of human neuroanatomy. In this case, the challenge is the brainstem and knowing where these cranial nerves 3 through 12 attach. Well, I want to lead by giving you a table. This is from our textbook, for those of you that have the textbook. If you don't, you can follow along in the handout that I've made available to you, or simply follow along on this chart. And what I've listed for you here in this chart are the 12 nerves that traditionally are called cranial nerves. And I just want to briefly walk through each one and then show you with some graphics how they relate to the brainstem itself. And then finally, we'll have a bit of a break, and you'll see me next in the lab with a human brain in my hand, and I'll show you these nerves as they actually present in a human brain specimen. And I'll also make use of a brainstem model that we have that will help clarify some of the nerves that are more difficult to retain in the autopsy specimen. Okay, so let's begin with our cranial nerves. So it's conventional that we identify cranial nerve 1 as a cranial nerve. It's the olfactory nerve. Cranial nerve 1, however, is not actually what we see on the bottom of the brain. Cranial nerve 1 are the axons that penetrate through the anterior floor of the cranium in a region called the cribriform plate, tiny axons that run from the nasal epithelium, where our sensory cells live, through the cribriform plate, and they make synaptic connections in the brain. And that structure in the brain is called the olfactory bulb. So when you see the olfactory bulb, don't mistake it for a cranial nerve. Know that that's part of the brain, but it's the part that receives cranial nerve 1. Okay, cranial nerve 2 is the optic nerve. So the optic nerve, very important nerve. It communicates visual signals from the retina back to the rest of the brain. And it's also a bit unusual. We call this a nerve, but in truth, it's an outgrowth of the diencephalon. As the brain develops, something called the eye cup forms off the lateral margins of the diencephalon, and this continues to grow outward and forms the neural elements of the retina. So in truth, the retina and the optic nerve are an extension of the brain. So we might think that for that reason we wouldn't call this a nerve, since it's not a true nerve in the conventional sense. But in keeping with our tradition in the nomenclature of these nerves, in medical neuroscience, we will in fact call cranial nerve 2 the optic nerve. All right, enough trick questions about cranial nerves. Cranial nerve 3 is the oculomotor nerve, and it's a very important motor nerve. And as we see in our table here, it's involved in mediating eye movements, as well as the motor arm of the pupillary light reflex. So we'll talk about all of this in more detail in a little bit when we get into the motor system. And also when we get into the visual system. We'll come back and spend quite a bit of time actually talking about nerves 2 and 3 in that context. Well, nerve 4 is a very small nerve, unusual in a few respects that I'll mention when we get there, but it's also a motor nerve and is involved in the control of eye movements. Now that brings us to the trigeminal nerve, nerve number 5. The trigeminal nerve is a very large nerve, very important nerve in the cranial region. It provides for somatic sensory signals to pass into the brain stem, and it also allows for motor signals to leave the brain stem to innervate the muscles that are involved in chewing. So the trigeminal nerve is involved in both somatic sensation from the face, mouth, as well as the cornea of the eye, but also in the muscles of chewing, and we call that mastication. Okay, nerve 6, the abducens nerve. This is the last of the 3 nerves that pertain to the movements of the eye within the orbit. So this is a motor nerve, and as its name implies, it abducts or abducts, moves lateral the eye within the orbit. Okay, now we're getting into some interesting nerves. I'm going to take nerves 7, 9, and 10 together. So nerves 7, 9,
10. We'll come back to 8 in just a moment. The facial nerve, the glossopharyngeal nerve, and the vagus nerve. So what I'd like to highlight is that each of these nerves is a mixture of both sensory and motor signals. So there's a sensory component to each of these and a motor component to each of these. So while on the face of it, these seem like the most complicated of all the cranial nerves, and indeed they are, but they're all complicated in the same way. They all have sensory components, they all have motor components. Now this table is not exhaustive, but I'll just highlight some of the principal functions of each of these nerves. What I want you to take away at this point for the facial nerve is that it's involved in providing our sense of taste from the anterior part of the tongue. So that's an important sensory component of the facial nerve. An important motor component is that it is involved in governing the muscles of facial expression. So next time you smile, or next time you are in a social situation and you're observing just the full range of facial expressions that we humans express, remember that it's the facial nerve that's motivating the contraction of those facial muscles. So facial nerve, muscles of facial expression, and taste from the anterior 2/3 of the tongue. Okay, nerves 9 and 10, again, very complicated nerves, complicated in the same way. Nerve 9 is the glossopharyngeal nerve. That tells you something about what it does. Glosso is a word that implies the tongue, and pharyngeal implies the pharynx. So it's a nerve that's related to the tongue and to the pharynx. It conveys sensory signals about taste from the tongue, along with nerve 7, and it's involved with providing motor output to muscles around the region of the larynx and the pharynx. And that's what we find here in our table here to the right. One additional function that's worth highlighting for the glossopharyngeal nerve is that it also provides important sensory signals that are important in cardiovascular control, specifically signals derived from the carotid bodies as well as the aortic arch. Okay, and nerve 10, the vagus nerve. Like glossopharyngeal, it's also complicated. It conveys sensory signals from the viscera, and it conveys a special sensory signal about taste. So 7, 9, and 10 are all concerned with taste, among other kinds of sensory signals that are coming into the brainstem. And in terms of the vagus nerve, you may already know something about this nerve. This is a nerve that's involved in providing parasympathetic outflow to viscera in the thorax and the upper part of the abdomen. It's also involved in innervating parts of the pharynx, including the vocal cords. So it's important in speech, it's important in swallowing. All right, so I'm going to jump back now and pick up cranial nerve 8. Cranial nerve 8 is the vestibulocochlear nerve. It's entirely sensory. It's a special sensory nerve. It communicates back to the brainstem our sense of hearing from the cochlea in the temporal bone, but also a sense of balance, a sense of the signals that are detected by the vestibular labyrinth in the temporal bone, which allows us to sense the tilt of the head as well as its rotational accelerations and linear accelerations. So these signals are very important in helping us maintain a sense of balance, as well as helping us to maintain our point of fixation, whatever it is we're looking at, even if we decide to move our head back and forth. So we'll see demonstrations of that in a little bit and talk more about that in detail. All right, so finally nerves 11 and 12. Nerve 11 is the spinal accessory nerve. The spinal accessory nerve is derived from the upper cervical segments of the spinal cord. It's an unusual nerve in that it is formed by rootlets that come off the upper part of the cervical spinal cord, enter the foramen magnum, so now it's in the cranium, and the nerve loops around and then exits the cranium again after picking up a small contribution from the brainstem. And then that nerve exits out of foramen with cranial nerves 9 and 10, the jugular foramen. So from that standpoint, it's fair enough to call it a cranial nerve, even though most of its origin is actually below the cranium. Well, nerve 12, the hypoglossal nerve, this is a nerve that controls the extrinsic muscles of the tongue. So if you stick your tongue out, so everyone stick your tongue out, straight out, well, that's your hypoglossal nerve in action. And hopefully your tongue went straight, because if it didn't go straight, if it deviated to one side or the other, that could be a very important clinical sign, and we'll spend some time talking about that one. All right, I think we're ready now to look at the brainstem. So, I think you've seen the brainstem already. If you haven't, you might want to back up just a bit and view my tour of the human brain that I provided in some of the earlier neuroanatomy tutorials. But for our purposes here, I want you to focus on some of the key features of each of the three main divisions of the brainstem so that then you'll know how these cranial nerves attach. So what we're looking at is a simplified illustration of the ventral surface of the brainstem with some additional structures attached up above. So we won't worry so much about what we see up in this region. That's actually part of the forebrain, including structures like the basal ganglia and the thalamus. So we don't have the olfactory bulbs here, so we're not going to see nerve 1, so we won't worry about that. I'll first just highlight this region, this junctional region between the caudal diencephalon and the midbrain. So this region shown in this purple color is in fact the midbrain, and we know that because of these broad stalks of white matter that we have on the ventrolateral aspect of the midbrain. That's called the cerebral peduncle. The space between the peduncle is called the interpeduncular fossa, and that's where we find cranial nerve 3. So here's cranial nerve 3 emerging from the interpeduncular fossa. Now, on the dorsal aspect of this brainstem, right near the junction of the midbrain and the pons, which is just inferior to the midbrain, we see a little bit of the fourth nerve peeking out from the dorsal aspect of the brainstem. There it is on the other side here. That's cranial nerve 4, the trochlear nerve. What will be important to remember about the trochlear nerve is that it's the only motor nerve that exits on the dorsal side of the central nervous system. All the other motor cranial nerves and the ventral roots of the spinal cord are all exiting on the ventral or anterior aspect. Okay, well, just below the midbrain, we have the pons. The pons is a large bulbous-like structure that is impossible to miss when you're looking at the ventral surface of the brainstem. And pons is one of those great words in neuroanatomy that's worth knowing something about. Pons means bridge. And the idea is that there's a long axis to the brainstem, and then the pons forms a bridge around this long axis of the brainstem at roughly a 90-degree angle. So the pons contains these axons that are running in the medial to lateral direction. And these axons, we'll learn more about them when we talk about the cerebellum. They're providing input to the cerebellum. So out here we have the cerebellum, and these axons are terminating, leaving the pons, terminating in the cerebellum through a structure that we'll call the middle cerebellar peduncle. Well, I'm getting ahead of myself. For now, I just want you to be able to recognize the pons as this bridge across the longitudinal axis of the brainstem. All right, well, as you can see from this illustration, once we get below the pons, things get real busy real fast with respect to cranial nerves. So I want you to notice the junctional region between the pons and the medulla oblongata, or just the medulla for short. So along this junction, we've got three pairs of cranial nerves. And below that junctional region, I want you to notice that the medulla has something of a longitudinal axis to it. There's sort of a series of bulges and furrows in the longitudinal axis. So that defines some locations where I want you to look for certain cranial nerves. The medial of these two bulges is very important. We'll spend a lot of time talking about those structures. That's the medullary pyramid. That's the cortical spinal tract that allows the cortex to control the actions of the spinal cord. Just to the lateral side of that medullary pyramid is a really lovely bulging structure, which is called the olive. It sort of looks like an olive cut in the long dimension of that fruit and then stuck to the side of the brainstem. That's the olive. So we're going to look for cranial nerves between the medullary pyramid and the olive, and then we'll look for cranial nerves that are just lateral to the olive. All right, well, let's go ahead and look at a similar illustration of the ventral surface of the brainstem, now with all the nerves attached and labeled and color-coded as to whether it is a purely sensory nerve, a purely motor nerve, or a nerve that has both sensory axons and motor axons. So again, ignoring nerve 1, which is in the anterior portion of the cranial floor, we have the optic nerve, which comes together just in the anterior part of the hypothalamic region, the base of the diencephalon. We see the nerves coming together just in front of a structure called the infundibulum, or the stalk of the pituitary gland. We'll see that nicely in the laboratory. Well, also just behind this region, we have the mammillary bodies, an obvious feature of the posterior floor of the hypothalamus. When you see the mammillary bodies, you know you're about to transition into the midbrain, and that would be also indicated by the prominent cerebral peduncles. And so now we know to look for nerves 3 on the ventral surface and nerve 4 emerging from the dorsal aspect of the midbrain. Well, the pons is what's next, going in the inferior direction, and plunging right through these transverse fibers of the pons is the trigeminal nerve. So that's an easy one to find. You'll know where to look for that. It's the only cranial nerve that seems to be emerging from the ventrolateral aspects of that beautiful bridge across the long axis of the brainstem. Well, that brings us to the junctional region between the pons and the medulla, where we find cranial nerves 6, 7, and 8. And they are in medial to lateral sequence, with nerve 6, the abducens nerve, close to the midline. And then some distance out, we encounter nerves 7 and 8, which are actually rather close together. This illustration makes them look like they're sort of evenly spaced, but they're really not. Nerve 7 is right on the medial side of nerve 8, with nerve 8 being the most lateral. Now, if we eye the place where we find cranial nerve 8, we'll find nerves 9 and 10 just below cranial nerve 8. So the glossopharyngeal nerve is this one right here, and then just below it are a series of nerve rootlets that comprise the rootlets of the vagus nerve, nerve number 10. And then roughly in the same position with respect to the lateral aspect of the lower part of the medulla, we find the spinal accessory nerve, nerve number 11. And as I mentioned, there are rootlets that gather up from the upper cervical segments of the spinal cord. They form a nerve that then enters through the foramen magnum, which is roughly at about this position here. So there's our foramen magnum at the base of the skull. And this nerve enters then the cranial vault, and it receives a small contribution from the medulla, and together with nerves 9 and 10, it exits the cranium via the jugular foramen. So in that sense, it's a real cranial nerve. And then lastly, the hypoglossal nerve. I mentioned the medial medullary pyramids right along the midline of the medulla. Here they are here. And then just to the lateral aspect of the medullary pyramids is this olivary structure, the inferior olive. And between the pyramid and the olive, then, that's where we find our hypoglossal nerve. So it's hard to see in real brains. After they've been removed from the skull, often these tiny rootlets will tear. But hopefully I'll be able to give you a view of the hypoglossal nerve roots in a brain specimen at some point during this course. Okay, well, let's briefly just talk a little bit about the spinal cord. There's just a few points that I want you to take away from this discussion of the spinal cord. One is that there is a longitudinal organization to the spinal cord, which is to say there are segments of the spinal cord that can be recognized by the attachment of 31 pairs of spinal nerves. So in the cervical region of the spinal cord, we have 8 pairs of nerves that attach. In the thoracic region of the spinal cord, we have 12 pairs of nerves that attach, one on either side, of course. And then both in the lumbar region and in the sacral region of the cord, we have 5 nerves that attach, 5 lumbar and 5 sacral. And then finally, there's a single coccygeal nerve that attaches at the very bottom of the spinal cord. So when you add all those numbers up together, we get the number 31. So there are 31 pairs of spinal nerves, 8 cervical, 12 thoracic, 5 lumbar, 5 sacral, and 1 coccygeal. Now, that's one way of characterizing the long axis of the spinal cord. Another way is to note that there are 2 regions where the diameter of the spinal cord is enlarged. One in the cervical region and another in the lumbosacral region. Now, you can probably imagine what's going on here. There's an expansion of the numbers of neurons and the circuitry of the spinal cord that's related to these important locations along the vertebral axis of the body. Namely, this is where our arms are attaching to the body axis and the legs. And in these 2 regions of the spinal cord, these regions are where we find the nerves that are providing motor output to the extremities as well as providing sensory input. So there are enlargements or an increase in the diameter of the spinal cord. There in the sacral region, in the cervical region, as well as in the lumbosacral region. And this thickening then allows for an increase in the circuitry that is required to govern the activities of the arms and the legs. One other point to make before we leave this view of the spinal cord, I want you to notice where we find the location of the inferior end of the spinal cord. We find it not at the bottom of the vertebral column, but we actually find it at about the level of L1 or L2, that is vertebral body levels L1 or L2. Well, of course, it didn't begin that way. As the spinal cord was first developing, it was approximately the same length as the bones of the vertebral column. So the spinal cord filled up that space within the vertebral column. But as we grow, and especially in early life, the non-neural structures grow at a faster rate, so we end up with a longer vertebral column than spinal cord. And as a result, the spinal nerves end up continuing to exit through the canals where they first developed, but now there is a long length of nerve root that develops as the vertebral column grows at a faster rate than the spinal cord. And what we end up with as a result of all of this is a beautiful structure called the horse's tail, or the cauda equina. And so in the lumbosacral regions of the spinal cord, what we see are these long nerve roots that extend like a horse's tail from the lumbosacral enlargement out through the canals between the vertebrae, space called the intervertebral foramina, that allow for the passage of these spinal nerve roots. So I say all that to emphasize that I don't want you to be confused about the nomenclature that we use to describe level within the spinal cord. If we're talking about, for example, the L2 level, you need to understand, are we talking about the spinal nerve? Are we talking about the vertebra or the vertebrae itself? Or are we referring to the segment of the spinal cord? And I'll always try to be clear about that as we're talking about these anatomical issues. So for example, the second vertebral body is found right at this level here. And that's where we find the emergence of the second lumbar nerve. However, that nerve attaches to the spinal cord at some significantly higher level. So when it comes to understanding the relationship of lesions within the spinal cord to dermatomal levels of paresthesia or anesthesia, it will be important to know these relationships between spinal cord segment, nerve, and vertebrae. So I'd just like to make a few points as we look a little bit more closely here at the vertebral column. What I want you to notice is that the dorsal or the posterior aspect of the vertebrae is off to the right, sort of coming at you, and the ventral or the anterior aspect is to the left, with the dorsal ventral axis running something like this through this illustration. Okay? So then it makes sense to recognize a ventral root that's coming out of the ventral horn of the spinal cord. This ventral root is going to send signals out to striated muscle, and these are motor signals leading to the contraction of those muscle fibers. Now, that spinal nerve is also going to contain sensory axons that may be derived from special receptors in skin or some other kind of structure, and the course of these axons will run right through that same spinal nerve, so we call that a mixed spinal nerve. And then there's a cell body that sits in a ganglion right here, and the central process of that axon will enter the spinal cord. So when we're talking about the spinal nerve itself, I want you to understand that it is mixed with both motor and sensory components. But once we get down to the roots, then we have a ventral motor root and a dorsal sensory root. All right. Now we also have something called the sympathetic chain, which is a chain of ganglia that runs in the long axis, or the superior to inferior axis of the spinal cord on either side. And we'll talk about this when we get into a discussion of the autonomic nervous system. But within these sympathetic chain ganglia are the neurons that innervate viscera, innervate vascular arterioles, basically wherever there is sympathetic innervation of smooth muscle, cardiac muscle, and glandular tissue. But that's where we find those kinds of motor neurons, in the sympathetic chain. And notice that the sympathetic chain is connected by a series of small connecting rootlets that go back and forth between the spinal nerve and the sympathetic chain as that nerve emerges out of the canal formed between the vertebrae of the vertebral column. All right, well, that's really all that I wanted to tell you today about cranial nerves and spinal nerves before having a chance to focus in on the brainstem and show you the cranial nerves in the lab. So if you'll bear with me, we'll make a transition. And when you see me next, I'll be in the neuroanatomy lab with the brain in my hands. So I'll see you there.
For this lab lesson, I want to show you the cranial nerves in a human brain specimen. So there are 12 nerves that attach to the brain and the brain stem, and most of them will be visible in this specimen, but perhaps we want to appeal to a model of the brain to show you one that often is lost in the autopsy room when we're removing the brain from the cadaver. But with that exception, let's begin with cranial nerve 1. Now, cranial nerve 1 is actually not present in the brain because it is a set of fine axons that run from the nasal epithelium to this structure right here called the olfactory bulb. And so the olfactory bulb is actually part of the brain, even though the bulb itself and its long connection that goes back to the posterior part of the frontal lobe looks very much like a nerve. It really is not a nerve, it's part of the brain. It's derived in embryogenesis from the telencephalon. So cranial nerve 1 would be those axons that run through the ethmoid bone through a region called the cribriform plate and make synaptic connections here with the olfactory bulb. Cranial nerve 2 is the optic nerve, and that is a very prominent nerve here in the human brain, and you'll notice that there are two. In fact, there are two of each of these nerves, but in this case, these two nerves seem to come together. So this is the right optic nerve, cranial nerve 2, the left optic nerve, and where they join, they form something of an X shape, and that X shape is called the optic chiasm. So just behind the optic chiasm is this stalk that extends from the hypothalamus down below the brain. Now this might also look like a nerve, but it's not. This is the stalk of the pituitary, called the infundibulum. And it's worth noting the proximity of this infundibulum to the optic chiasm. Unfortunately, one problem with the pituitary is that sometimes it tends to grow tumors. And because the pituitary is encased in bone, should a tumor develop in the pituitary, the only place for it to grow is upward into the optic chiasm. So sometimes, for that reason, visual field deficits are the first and earliest sign of a problem in the pituitary region. So cranial nerve 2, the optic nerves that come together to form the optic chiasm. Now, in a different lesson, we'll talk about the blood vessels. Just behind what is a particular vessel called the posterior communicating artery is the third cranial nerve. Here's the left third cranial nerve, and here's the right. This nerve is also called the oculomotor nerve. It supplies many of the axons that drive the movements of the eyes. Now, in order to see the fourth cranial nerve, we need to gently reflect away part of the cerebellum and brainstem so that we can peer deep down beside the pons and just below the temporal lobe. The fourth cranial nerve is called the trochlear nerve, and it's unusual for two reasons. One is that it's the only motor nerve that exits on the posterior or the dorsal side of the central nervous system. So to see the trochlear nerve, we have to gently reflect away the cerebellum from the inferior surface of the temporal lobe. And this fine little thread structure right here at the tip of my pointer, this is the trochlear nerve. The trochlear nerve exits on the dorsal aspect of the caudal midbrain, wraps around the brainstem, and works its way towards the orbit, where this nerve is involved in controlling one particular muscle that moves the eye downward and inward. Now, the other unusual aspect of this trochlear nerve is that it's the only motor nerve that supplies muscle that is on the opposite side of the body from the source of this nerve, which is to say, I'm holding at the tip of my wooden stick here the right trochlear nerve. This trochlear nerve was grown out by motor neurons that sit on the left side of the brainstem in the trochlear motor nucleus of the caudal midbrain. So that is cranial nerve 4. Cranial nerve 5 is a large nerve that penetrates right through the lateral aspect of the pons. And this nerve has been cut fairly close to the pons, so we don't have a long length to show you, but I think you can appreciate this tuft of nerve fibers that's right at the end of the pointer here, that's penetrating down through the axons that connect the pons to the cerebellum. That's the fifth cranial nerve, the trigeminal nerve. Cranial nerve 6 is found right near the junction of the pons and the medulla oblongata. This, along with cranial nerves 3 and 4, is a nerve involved in moving the eyes. This is the abducens nerve. It's the most medial of 3 nerves that are present right along the junction of the pons and the medulla. So that's cranial nerve 6. Cranial nerves 7 and 8 are out further laterally, still at the junction of the pons and the medulla. Cranial nerve 7 is right here. This is called the facial nerve. It's one of our most complicated nerves in that it has both motor and sensory fibers associated with it that do a variety of functions that we'll describe in a different lesson. Now, just lateral to the facial nerve is cranial nerve 8, the vestibulocochlear nerve. And as its name implies, this nerve is involved in both vestibular function, related to our sense of balance, and auditory function, related to our sense of hearing. Nerve 8 sits in the lateral margin of this junction between the cerebellum, the pons, and the medulla. Just below nerve 8 are nerves 9 and 10. Nerve 9 is called the glossopharyngeal nerve. And here it is right here. It sits right below nerve 8. And then just below the glossopharyngeal nerve are the nerve rootlets of the vagus nerve, which is cranial nerve 10. And we see a collection of these nerves that are emerging from the lateral margin of the upper medulla, cranial nerve 10. Now, this particular specimen has been cut through the upper part of the medulla, and in the medulla is where we would find the nerve roots of our last two cranial nerves, nerve 11 and 12. So to show you those nerves, I'm going to appeal to a model of the brainstem that I have that will give you a sense of how those nerves relate to the medulla. In order to see cranial nerves 11 and 12, I'm going to appeal to the brainstem model that I have here in my left hand. And I'm holding the brainstem model next to the actual brain so that you can get oriented and recognize the divisions of the brainstem that are depicted here in this model. Near the middle we have the pons. Above it you can see the cerebral peduncles, which are a hallmark of the midbrain. And then down below the pons is the medulla oblongata, which in the model is depicted to extend into the spinal cord. So let me put the human brain to the side, and we can focus our attention on the medulla in the model so that we can observe cranial nerves 11 and 12. Cranial nerve 11 is this long flanking structure that emerges from the lateral side of the upper cervical spinal cord and the caudal part of the medulla. This is called the spinal accessory nerve. It's an unusual nerve in that it includes contributions throughout the upper cervical spinal cord, and it enters the cranium through the foramen magnum, only to loop back out through the jugular foramen. So it may be somewhat questionable whether we should think of this as a cranial nerve, but the fact that it does enter the cranium is good enough reason to include it in our standard means of accounting for 12 cranial nerves. So cranial nerve 11, the spinal accessory nerve. Cranial nerve 12, unfortunately, was not present in our brain specimen because there are tiny rootlets that often tear in the process of removing the brain from the skull, which was the case in our specimen. But in the model, cranial nerve 12 is represented by this series of rootlets that emerge right next to the olive, which is this lateral bulge of the upper part of the medulla, and the medullary pyramid, this long column that runs along the midline of the ventral medulla. So the proximity of the nerve rootlets of the hypoglossal nerve to the medullary pyramid is worth remembering. The medullary pyramids contain axons of the cortical spinal tract, allowing the motor centers in the forebrain to communicate with the spinal cord. So right next to it are the rootlets of the 12th cranial nerve, the hypoglossal nerve, which is the nerve that allows us to protrude our tongue. This nerve innervates the intrinsic muscles of the tongue. Unfortunately, some patients have strokes or other kinds of brain injuries that impact this region in the medulla. And in such patients, there is a very predictable set of neurological signs and symptoms. Because of the cortical spinal fibers in the medullary pyramid, those patients are weak on the opposite side of the body. But because of involvement of the hypoglossal nerve roots, those patients also have problems extruding their tongue. And specifically, the tongue will deviate towards the side of the injury because of the architecture of the fibers of the intrinsic muscles of the tongue. So a deviating tongue and weakness on one side of the body are hallmarks of injury to this upper ventral medulla, where the roots of the hypoglossal nerve penetrate right lateral to the medullary pyramid. So this model allows us to account for all 12 cranial nerves. And just to show you one more time, we have the olfactory nerve connecting to the olfactory bulb. That nerve is really in the skull. And then nerve number 2 is the optic nerve. Nerve number 3, the ocular motor nerves that leave just to the medial side of these cerebral peduncles. Nerve number 4 wraps around from the dorsal aspect
of the caudal midbrain. That's the trochlear nerve.
Nerve number 5 is the trigeminal nerve that penetrates right through these transverse fibers of the pons. And then at the junction of the pons and the medulla, we have 3 pairs of nerves, the abducens, the facial, and the vestibulocochlear nerve, nerves 6, 7, and 8. And then just below cranial nerve 8, we have the glossopharyngeal nerve, nerve number 9. Just below that are the rootlets of the vagus nerve, nerve number 10. And then coming up from the cervical spinal cord is the spinal accessory nerve. And then lastly, the hypoglossal nerve right between the olive and the medullary pyramid.
Hello everyone, welcome again to my home, and today I'd like to talk to you about the function of the cranial nerves. This topic relates to one of our core concepts in the field of neuroscience. It's core concept number 1, that the brain is the body's most complex organ. And the complexity we're going to explore today concerns the functions of the 12 cranial nerves. My learning objectives for you today are first, that you would discuss the functions of the cranial nerves in terms of the sensory and motor signals that are conveyed by each nerve. Some of them will convey only sensory signals, some just motor signals, and others both. I also want you to be able to discuss the relationship between the cranial nerves and the corresponding nuclei within the brainstem that either receive signals from those cranial nerves or give rise to axons that grow out and form those nerves. Well, in a previous tutorial, you've had the chance to look at the cranial nerves on the actual human brainstem, as well as to study them, I trust, in this illustration from our textbook that we've been reading along the way. This is from the appendix, figure A7, and this figure lays out for you the cranial nerves that attach to the brainstem. There's no olfactory nerve here because the olfactory nerve does not attach to the brainstem, but we do have our optic nerve, which attaches actually to the forebrain, to the diencephalon, not to the brainstem, but it's included in this illustration because of its proximity to the brainstem. And then the rest of these nerves, 3 through 12, do indeed attach to the brainstem. They are the ocular motor nerve, number 3, the trochlear nerve, number 4, the trigeminal nerve, number 5, the abducens nerve, number 6, the facial nerve, the vestibulocochlear nerve, nerves number 7 and 8, the glossopharyngeal nerve, the vagus nerve, the spinal accessory nerve, and finally the hypoglossal nerve. So if you need a visual reminder, I would point you back to the tutorial that was presented in week 1 that refers you to the anatomy of the brain stem as viewed in an actual human specimen. And there you can find a couple of different tutorials that identify these cranial nerves as they attach to the brain stem. Part of our challenge that we're building towards with this tutorial is to try to get to the point where we're able to relate those nerves to the set of gray matter structures within the brainstem that either receive the incoming sensory signals or give rise to outgoing motor signals. Now, I know this figure must look like a monster to you, and indeed it's rather onerous, but it's an attempt to represent for you the 16 or 18, depending upon how you want to count them up, nuclei within the brainstem that are related to these cranial nerves. So we're not going to take this challenge on full bore right now. We'll do that progressively over a few tutorials. Rather, what I want to be able to do for you is give you a way to understand the function of the nerves so that we can then get into the brainstem and recognize how the gray matter nuclei help to explain the functional differentiation of the various components of each nerve. So here's how I'd like to do that. I'd like to build for you out this table. And in this table, I've organized the content, that is the function of the cranial nerves, into motor components and sensory components. Now, notice that I'm focusing on just nerves 3 through 12. So what I'm leaving off here is nerves 1 and 2. Those are the two nerves that don't attach to the brainstem. Just for completeness, let me tell you what they are now, and then we won't return to them. Nerve 1 is the olfactory nerve. These are the axons that grow from our olfactory sensory cells in the nasal epithelium through the cribriform plate at the anterior floor of the cranium and make synaptic connections into the olfactory bulb. That's nerve 1. Nerve 2 is the optic nerve. The optic nerve grows out from retinal ganglion cells. These are the neurons in the inner layer of the retina that extend their axons back to the diencephalon and to the very upper part of the midbrain. We'll get back and talk more about that later. Okay, so nerves 3 through 12, these are the ones that fully attach to the brainstem. And notice how they've been further differentiated. On the motor side, we've recognized 3 divisions of motor outflow, which is a reflection of the embryology of the developing nervous system in relation to the developing targets of these motor axons. We can recognize somatic motor nuclei. So these are nuclei that contain the motor neurons that innervate the muscles that are derived from embryological somites. We have branchial motor nuclei. These are nuclei that contain motor neurons that grow out and innervate the muscles derived from the pharyngeal arches in the developing embryos. These are called branchiomeres, and from those branchiomeres develop a set of structures in the cranial region and in the neck. Some of them are muscle, and some of those muscles are innervated by nuclei that come to occupy a distinct position in the brainstem. So these we call our branchial motor nuclei. And then lastly, on the motor side, we have visceral motor nuclei in the brainstem. And so these are the preganglionic parasympathetic neurons that provide outflow from the central nervous system to ganglia associated with end visceral organs. And then from there, there's a ganglionic neuron that actually innervates the end target, be it smooth muscle or cardiac muscle. Well, that's on the motor side. On the sensory side, we have subdivisions that will also help us to organize our understanding of these nerves and their functions, and eventually how that relates back to the brainstem. There is a special visceral sensory function associated with some of the cranial nerves, and that function is taste, or a little bit more technically, gustation. So we want to recognize which nerves convey our sense of taste to the brain. There's a more general visceral sensation that comes from some cells that are associated with the carotid body as well as the aortic arch. And depending upon how we want to think about that, we can also consider the mucous lining of the oral pharynx as part of this general visceral sensation. Okay, we have next somatic sensation, which would include those structures that are in the peripheral parts of our body, namely our skin surfaces, our muscles, our joints, our tendons. So from these somatic structures are derived sensory signals that feed into the brainstem, specifically from the region of the head and the neck. And then lastly, in the cranial region, we have special sensory systems. And there are some that attach to the brainstem, specifically first-order afferents that are concerned with our sense of hearing and our sense of balance. Okay, so this is the embryological framework that we're going to apply. What I'd like to do next is just walk through nerves 3 through 12 and identify which components we find associated with each. So beginning with nerve 3, our ocular motor nerve. What we have in the ocular motor nerve is a somatic motor function and a visceral motor function. The somatic motor function of nerve 3, the ocular motor nerve, concerns the outflow to the orbital muscles that move the eyes, and also to a muscle that elevates the eyelid. On the visceral motor side, the outflow through the third nerve is a parasympathetic preganglionic outflow that's involved in constricting the pupil. We'll come back and talk more about that when we consider the pupillary light reflex. Okay, nerve 4 is the trochlear nerve. It has a single component. It's a somatic motor outflow. The trochlear nerve innervates the superior oblique muscle, one of those muscles in the orbit. This is a muscle that turns the eye inward and downward. Nerve 5 has two components. It has a branchial motor component. It supplies motor outflow to the muscles of mastication. That is, our chewing muscles. So these are muscles that are derived from the pharyngeal arches. There's also a sensory component in nerve 5. This is a general somatic sensory component derived from much of the face. And our sense of touch, our sense of movement of the face, this is conveyed via the trigeminal nerve, in addition to some others, but the trigeminal nerve is the most significant of the cranial nerves, providing incoming general somatic sensation. Nerve 6, like nerve 4, only provides a single somatic motor output to one muscle in the orbit. And nerve 6 is the abducens nerve, and the muscle it innervates is the lateral rectus, which abducts or abducts the eye, moving the eye laterally. Now, nerve 7 is one of the more complicated nerves that we have, and as you'll see, there are numerous components to nerve 7, so let's talk about them in turn. First, with the branchial motor component. This is perhaps the most memorable. So nerve 7 is the facial nerve. It is the nerve that supplies the muscles of facial expression. So those muscles are derived from the pharyngeal arches, and it is branchiomeric. There is also a parasympathetic outflow to numerous glands in the cranial region, the lacrimal glands as well as the salivary glands, except for the parotid, which is supplied by a different nerve. So that's the parasympathetic outflow from the facial nerve. There's a special visceral signal coming from the anterior two-thirds of the tongue that passes through the facial nerve. So this is information about taste or gustation conveyed via the facial nerve. And then there is a general somatic sensory component derived from a small patch of skin in the ear. Okay, nerve 8 is a sensory nerve. And I have two circles way off to the right-hand column for special sensation for nerve 8, because there are really two components that are passed through this nerve. There's a hearing component conveyed from the cochlea, and a balance or equilibrium component derived from the vestibular labyrinth. And it's worth putting two check marks separately here in this box for nerve 8, because these two divisions of the nerve will innervate different sets of nuclei in the brainstem. Okay, nerve 9, the glossopharyngeal, is one of the most complicated nerves that we have. Even one more check than what we had for nerve 7. So again, let's walk through the glossopharyngeal systematically, beginning with the branchial motor component. So as the name implies, there's innervation of muscles around the pharynx out from nerve 9. That gives us the pharyngeal component of the glossopharyngeal. There is a parasympathetic outflow in nerve 9 to the parotid gland, one of our salivary glands. There's a special visceral sense that's derived from the posterior 1/3 or so of the tongue. So this is our sense of taste being conveyed from taste buds in that posterior region of the tongue. There is a interesting general visceral sensory signal that is conveyed via the glossopharyngeal nerve. And this is coming from the carotid bodies, which are chemosensory structures associated with the bifurcation of the common carotid into its internal and external parts. And so this is very important in regulating blood pressure. The carotid bodies are important for sensing the chemical composition of the blood, which then can feed back into the regulation of blood pressure, of cardiac output, as well as respiration rate. So these signals feed into the brainstem via the glossopharyngeal nerve, and this is what we have in mind when we talk about our visceral sensory element here. Now there's a somatic sensory component as well, also derived from a small patch of skin in the external ear, and that accounts for these 5 components of the glossopharyngeal. Now, the next nerve we have is the vagus nerve. Like the glossopharyngeal, it also has 5 components. And beginning with the branchiomotor component, the vagus provides some innervation to muscles in the posterior pharynx and in the region of the larynx. There's a parasympathetic outflow in the vagus nerve, of course. This is the one that you're probably thinking about when you think about the vagus nerve. There's outflow to viscera in the thorax and in the upper part of the abdomen. And much of that outflow is derived from a couple of nuclei in the brainstem. There is also a sense of taste that's conveyed via the vagus nerve. It's not quite as important as nerves 7 and 9, but it's there. It's from some taste buds that are in the posterior part of the oral cavity. There is a visceral sensory signal conveyed via the vagus nerve. It also has to do with regulating the cardiovascular system, and it has to do with sending signals from the region of the aortic arch that are sensitive to stretch, as well as sensitive to the chemical composition of the blood in that region. And lastly, like nerves 7 and 9, there's some innervation to a patch of skin around the external auditory meatus. So I want you to notice that nerves 7, 9, and 10 are all very complicated nerves. They're mixed nerves. They're mixed with multiple subcomponents for motor control and sensory processing. But they are complicated in very similar ways. So hopefully that will help you in your understanding of these nerves. All right, so the last 2 are a lot simpler. Nerve 11 is the spinal accessory nerve. It also has 2 check marks because this nerve provides for branchiomotor output to 2 different muscles, the upper part of the trapezius muscle and the sternocleidomastoid muscle. These are muscles involved in turning the head and shrugging the shoulders. And then lastly, the hypoglossal nerve, nerve 12, supplies for a somatic motor output to the tongue muscles, and this is the nerve that's involved in sticking your tongue out. All right, so there are a couple of dimensions in which I would hope that you would use this table in your studies. We've been emphasizing the horizontal dimension, that is, looking one nerve at a time, thinking about what are the various components that we would find in that nerve. So that's one way to approach using this table. Another would be the vertical dimension, and that is to think about, okay, what are the somatic motor components in the brainstem? And just think about the nerves, think about their anatomy, how they attach to the brainstem, and I would suggest that you do so thinking from superior to inferior. All right, so march through each column that way, the somatic motor, the branchiomotor, the visceral motor, and then on the sensory side, think about taste. How is taste conveyed to the brainstem, and so on. And I think you'll find that this table is not quite as daunting as it might first appear. Well, that's good, because when we get to the actual anatomy of the nuclei in the brainstem that connect to these nerves, it will seem a bit overwhelming. So I might ask you to brace yourself for that, but we can get through that together, and hopefully this chart in front of you will be one way to organize your understanding of these nuclei by name and by location, as well as by function. Okay, well, I'm going to try to simplify this for you just a little bit, borrowing a scheme that I've learned from my colleague here at Duke, Dr. Nell Kant. Dr. Kant suggests that we can think of the cranial nerves as being related to 16 cranial nerve nuclei. And these 16 nuclei can be grouped into 4 groups of 4. So it's sort of a 4 by 4 equals 16 perspective on the cranial nerve nuclei. So 16 cranial nerve nuclei that are subdivided into 4 groups of 4. 3 of these groups are motor nuclei, so that accounts for 12 of the 16 nuclei. There are 4 somatic motor nuclei, 4 branchial motor nuclei, and 4 parasympathetic motor nuclei to talk about. Well, that accounts for 12 of the 16, so that must mean that there is one group of sensory nuclei, and actually that group can be subdivided further according to the chart we saw on the previous slide. But to make this 4 by 4 framework stick, I think it might be helpful just to avoid thinking about that subdivision at the moment and consider 4 groups of 4 nuclei, 3 motor and then 1 group of sensory nuclei. Okay, now let's see how that 4 by 4 scheme maps now onto this chart where we've just gone through the cranial nerves that attach to the brainstem and identified the functions associated with each. So what we're going to do is talk about the nerves, but we're going to deal with them one column at a time. Okay? So let's go ahead and do so. The third nerve conveys somatic motor output to muscles in the orbit that move the eyes, as I suggested, and that output is coming from a nucleus called the ocular motor nucleus. The next somatic motor nucleus that we encounter is just a little bit inferior in the caudal part of the midbrain, and that's associated with the 4th nerve called the trochlear nerve. So 2 somatic motor nuclei in the midbrain, ocular motor superior and trochlear just inferior. Now the next somatic motor nucleus we have is in the caudal pons, and it's associated with the 6th nerve. It's the abducens nucleus. And then the final somatic motor nucleus, the 4th of that first group, is down in the medulla, in the upper part of the medulla associated with the roots of the 12th nerve, and it is called the hypoglossal nucleus. So there's our first group. There's our first 4 nuclei associated with somatic motor control. Now, for branchial motor, we have the trigeminal motor nucleus. It's the motor nucleus that grows out the motor axons in the trigeminal nerve. And this supplies for motor output to the muscles of chewing or of mastication. The 7th nerve, the nucleus there is called the facial nucleus. And that nucleus is responsible for governing the muscles of facial expression. Okay, so far so good. We've identified 6 cranial nerve nuclei, and the names of these nuclei all match the nerve that they contribute to. Well, that won't always be the case, as we're about to see. The branchial motor component in nerves 9 and 10 comes from a nucleus appropriately named called the nucleus ambiguus. It's very difficult to see in our standard histological preparation of the brainstem, which resembles a myelin stain. Of course, if we stained for the presence of cell bodies, especially for motor neurons, we'd see the nucleus ambiguus quite clearly, but usually histologists look at myelin stained sections through the brainstem, and it's quite obscure. So that's the nucleus ambiguus supplying branchial motor outflow through nerves 9 and 10. The branchial motor outflow through nerve 11 comes from the accessory nucleus in the upper cervical segments of the spinal cord. All right, what about the 4 parasympathetic nuclei? Well, beginning with nerve 3, there is a contribution that is responsible for the constriction of the iris. That comes from a nucleus called the Ettinger-Westphal nucleus, which sits just superior and a little bit lateral to the ocular motor nuclei up in the midbrain at the level of the 3rd cranial nerve. The next place where we have parasympathetic outflow is associated with nerves 7 and 9. And that outflow comes from a set of nuclei that we call the salivatory nuclei. Now we could subdivide them into a superior, a superior and an inferior group, but to keep this 4x4 scheme going, we'll just collectively consider them the salivatory nuclei. Okay, well, the next major parasympathetic outflow to consider comes from two nuclei. A division of the nucleus ambiguus, which is separate from the one that provides branchiomotor outflow. This division is responsible for the cardioinhibitory output that goes to the heart from the vagus nerve. Well, the other major parasympathetic outflow to consider in the vagus nerve comes from a nucleus called the dorsal motor nucleus of the vagus nerve. All right, well, that accounts for our three groups of motor nuclei. So let's just make sure we've got them all here. So that's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Okay, so we've got three groups of 4 motor nuclei. All right. Now on the sensory side, okay, we have 3 nerves in the brainstem that convey a sense of taste back into the central nervous system. They all supply the same nucleus. Actually, they supply the rostral division of this nucleus. The caudal division of the nucleus is the recipient of the general visceral sensations that are involved in regulating our cardiac rhythms, our blood pressure, and our respiratory rhythms. And that nucleus is called the nucleus of the solitary tract. The solitary tract is a very visually striking tract in the dorsal part of the tegmentum of the medulla, and there's a gray matter nucleus surrounding it. We call that the nucleus of the solitary tract. So notice there are 2 divisions of it, and as I mentioned, the rostral division is concerned with taste. The caudal division is concerned with visceral sensation. Our general somatic sensory system is again conveyed via multiple nerves, but to one nuclear complex, and that nuclear complex is the trigeminal nuclear complex. Now, when we get into our discussions of somatic sensation, including mechanosensation and pain and temperature sensitivity, we'll see that we can subdivide this trigeminal nuclear complex into 3 major parts, but we won't get into that now. We'll just recognize this as one nuclear complex. And then lastly, our special sensory nuclei in the brainstem include our cochlear nucleus and our vestibular nuclear complex. Okay, let's see how we've done here with our numbering scheme now. So we counted 12 motor nuclei. Here's one sensory nucleus. Here's a second sensory nucleus, and then a third and a fourth. Okay, so we've accounted for our 4 sensory nuclei to add to our 12 motor nuclei, giving us a total of 16. All right, well, I hope that scheme has helped you. We have lots of different ways to assist you in learning these nuclei and how to organize them. One additional method is shown in a slide that comes from the textbook that you may choose to read along with these tutorials. And this scheme is just a little bit different from the one that I showed you, but it still basically lays out subcategories for motor control and for sensation, and shows you how the nuclei can be arranged accordingly. Well, I hope this has been helpful. It's been sort of a whirlwind tour through the cranial nerves, emphasizing their functions, and then just giving you some handle on how those functions can be related to the nuclei of the brainstem. In a different tutorial, we'll actually see those nuclei and find them in histological sections. So that will be forthcoming, but before we get there, see if you can't rehearse a little bit in your own mind this 4x4 scheme for mapping the functions of the cranial nerves to the nuclei of the brainstem. I'll see you next time.